Now we're going to tie in what we learned about eukaryotic gene expression into differential gene expression in multicellular organisms. In other words, why an eyeball cell looks like an eyeball versus a muscle cell. They all have the same genes. How do they end up looking and behaving so differently? Now, there is a genetic program for embryonic development. We're going to kind of cover that along with cytoplasmic determinants and inductive signals. Then we're going to talk about sequential regulation of gene expression during cellular differentiation. So, you start out with the cell and it's fertilized. How does the cell know what ends up being the head versus the tail of this little baby frog, also known as a tadpole? Well, there's a pattern that starts from two sources. One are maternal signals. These are chemicals that the mom puts into the egg, and then when the sperm enters the cell, that gives the cell, the zygote, an orientation. So it can decide using those maternal signals plus where the sperm entered to decide what's up and what's down, what's the head and what's the tail. Then in the case of the tadpole, in 4 days you go from one cell into this multicellular organism that can eat algae and swim around and get away from predators. Now during this process, genes have to be turned on and turned off in the right order to go from a zygote to a multicellular individual. And the cells, to do this properly, not only rely on maternal signals that help them decide what's up, what's down, tail, head, but they also start talking to each other. Now through this process, cells start to differentiate. They become different. Genes are permanently turned on, genes are permanently turned off. Also, they do this by being induced by signals from the maternal signals that were in the egg, and also signals they start sending to each other. Okay, so let's talk about how some of this happens. The fertilized egg, the zygote, once it starts dividing, it reaches a stage in vertebrate development that's called the blastula, where you have a hollow ball. Then it starts to pinch in. Okay, because the cells are talking to each other, they start to migrate and they start to move. Okay, eventually this hollow ball that's now formed a U shape is going to form an organism that has a tube going from mouth to anus. Okay, depending upon where it pinched in. Then these cells that are on the outside, they realize that they're on the outside. They're only getting communication from one side, from this side. So they're going to turn into skin, neural tissue, and other organs. The middle part, because they're receiving signals from both sides all the way around, they're going to become muscles. The inside layer, okay, because it's only receiving information from one side, is going to become internal linings of organs, digestive organs, and that kind of thing. And it's all because they're talking to each other. And they say, okay, I ended up in the middle, therefore I'm going to turn off my skin genes and I'm going to turn on my muscle genes. And those skin genes eventually are going to be turned off permanently. Okay? Isn't that wonderful? Isn't that just kind of neat? Through the process of differentiation, we can see visible differences between the different types of cells. A muscle cell looks different than a skin cell, that looks different from a nerve cell, which looks different from a cornea cell. Okay, so there's differences in shapes, there's differences in cellular products. Okay, muscle cells produce different proteins than skin cells do, and also in some cases, some cells are told by the other cells that it's time to die. Going from the fin on that little tadpole, okay, to a limb with fingers, the cells in between the fingers have to die. That's probably more than you wanted to know. But it is very interesting, especially if you watch the clip that talks about the research that we're doing in organ generation. Really quite exciting. You don't have to watch it, but I would recommend it. It's in the supplemental section. Now let's talk about cancer and cell cycle control. This is where cells don't play nicely with each other anymore. Okay. Cancer cells have a tendency to look like undifferentiated cells, like the cells in the early embryo, okay, before they started differentiating into muscles and what have you. It's almost like somewhere along the line genes got turned back on that shouldn't have. Okay, so in our fight against cancer, in finding treatments for cancer that are better than what we've got now, they discovered genes in cancerous tumors, okay, that they called oncogenes. They thought that they were specific to cancer, but then as they got to looking, they found that oncogenes have analogs in non-cancerous cells. And these are genes that are generally related to the cell cycle, and they've become mutated so that they don't work properly, or they get moved to the wrong spot on the chromosome and they get turned on permanently, or they get turned off permanently, and you develop cancer. Let's talk about this in more detail. One of the proto-oncogenes that we've found is called RAS. You don't have to remember what a G protein is, but what happens is in the body, some cells will release growth factors that bind to a membrane receptor, which causes it to stimulate RAS. It talks to others, goes into the nucleus that activates a transcription factor, turns on a gene that promotes the cell, that causes the cell to divide. Okay, that's how it's supposed to act. Then when there's no growth factors, then the cell doesn't divide, or it divides more slowly. Well, if you get a mutation that causes this proto-oncogene to mutate into an oncogene or a cancer gene, it becomes hyperactive, and it does its job too well, which leads into too many of the genes that stimulate the cell cycle, and the cell divides without one being stimulated by growth factors, or two, it divides too many times from just one growth factor. So that's a case of a proto-oncogene mutating into an oncogene that stimulates the cell to divide too frequently. Now let's look at the opposite example. Okay, so say we've got some skin here, we've got UV light coming in, causes DNA damage. This stimulates signals that say, okay, let's turn on this proto-oncogene, okay, product called P53. It sits down on DNA and inhibits DNA replication until the damage can be repaired. Then we turn P53 off, then your skin cells are free to divide. Well, if you get a mutation so that you don't have the gene for P53, so you don't have the gene product P53 to stop the cell cycle when it needs to be stopped, you get mutations that get perpetrated by the cell dividing when it's got DNA damage, leading to mutations in the cells. Okay, that's another example of a proto-oncogene that halts the cell cycle versus the RAS gene that promotes dividing. Okay, now we're going to look at some animations that look at how proto-oncogenes can turn into oncogenes. We're also going to look at a clip on how cancer develops and spreads within the body.